Should get a little laser pointer here. Axon hillock, that's where action potential is generated. And now it needs to move down the length of the axon. The movement is the propagation. Researchers wanted to know, is this just one action potential that slides down the length of the axon? Or is this much more of a complicated process where the action potential needs to kind of replicate itself down the length of the axon? Turns out it is far more complicated than researchers thought. The action potential needs to replicate itself or repeat itself down the length of the axon. Probably takes energy. Enter myelin sheath. And it can actually just glide right over it. And then it gets to parts of naked axon, so now it's got to replicate itself again. And then it can glide over the segment of myelin. And then it's got to replicate itself a bunch of times, then it can just glide over. Do you see how important myelin sheath is now? Less energy. Makes it far easier to get across because it's not consuming all that energy. It doesn't have to replicate itself. It could just glide right over it. The more bare axon that shows, the more energy needed for it to replicate itself in those moments. That's how propagation occurs. What if the axon had no myelin sheath whatsoever? It would have to replicate itself the entire way down. Do you know how slow that would be? And do you know how much energy that would require? Neurons can do it. It would be slower and it would require far more energy. That axon was mostly myelinated. It's going to be quicker and it's going to require less energy. And so there's two types of conduction. There's one called passive conduction, has no myelin. This is not that there's anything wrong with it. It's going to require more energy and it's going to be relatively slow. The other one is called saltatory conduction. These are axons that have myelin. Segments of bare axon. They have a name. What are they called? Nodes of Ranvier. That is where the action potential needs to replicate itself or reproduce itself. Saltatory conduction, that action potential, even when it needs to reproduce itself over the nodes of Ranvier, still moves at about 268 miles an hour. Now we get to talk about the synapse. We talked a little bit about the synapse last week, but now that you know how the action potential is generated, we can kind of put the whole thing together. The presynaptic neuron. Again, my focus is right here, so what does this neuron become? Postsynaptic neuron, or the receiving neuron. You already know what type of conduction is going to occur here. Saltatory. Okay, so it should be quick. Space here, information is sent in what format? Chemical. Are you with me? What are those chemicals? Neurotransmitters. So now our job is to figure out how the neurotransmitters get spit out into the synapse. See here, little channels hanging out on the neural membrane. These neurotransmitters need to find their own openings. They need to find their docking stations. Different neurotransmitters have different shapes based on their chemical structures. Let's talk about how the neurotransmitters eventually get here, because it's a process. Do you remember when we talked about the neural cytoskeleton, we talked about there is a structure that helps to transport materials within the cell? So we have the microtubules that are really essential in moving the vesicles within the cell. These vesicles are spheres, and within those spheres are the neurotransmitters. And the microtubules help the vesicles to move down toward the axon terminal. The vesicles are going to move, they're going to fuse with the membrane, and then they're going to release their contents. So we've got this pattern of move, fuse, release. There's a name for this. It's called exocytosis. I do want you to know calcium is the ion that seems to, at this point, based on our knowledge, seems to govern exocytosis. The rest of the semester is built on this entire process. Once we're done with this, we can talk about different drugs and how they influence of the vesicles or how they sit in the gaps of the receptor sites of certain neurotransmitters. We can talk about how Parkinson's actually happens. We can talk about what Huntington's does. We can talk about what happens when someone has a stroke. And like if you understand chapter 3, you can understand the rest. Autoreceptors, which are found on the presynaptic neuron, seem to be in charge of understanding how much neurotransmitter is in the synapse, and then they let the presynaptic neuron know that it's done now. What happens to the neurotransmitter once released into the synapse? What are some of the options? What might some of these neurotransmitters do? Well, one thing that can happen is binding. This is when neurotransmitters find their appropriate receptor sites. They dock themselves, they transfer the information, that's binding. That sometimes happens. What's another thing that could occur? Reuptake. So the presynaptic neuron has the job of kind of reabsorbing or taking back some of the neurotransmitters that it released. The third thing that can happen to a neurotransmitter once it's released into the synapse is it could diffuse away. Okay, do you see this term, enzymatic breakdown? When neurotransmitters are spit out to the synapse, we have enzymes. Their job is to go into the synapse and break down neurotransmitters. So it's what we call enzymatic breakdown. A neurotransmitter that's broken down, or partially broken down, is not going to fit correctly in a receptor site. Neurotransmitters could bind, they go through a reuptake, they could just float away, or they could be broken down by enzymes. There's this optimal level of binding and reuptake and diffusion and enzymatic degradation that's needed in order for us to function to the extent that we do. So researchers have found that there are two types of receptors. One type of receptors are called ionotropic. The receptor site is actually on the ion channel. The right neurotransmitter finds its right receptor site. When that binds, then the channel opens. You get the rush of ions into the cell. So the whole point of binding is, in this case, it opens the ion channels, which gets us back to now potassium could rush in and trigger what? An action potential. So this is called ionotropic receptor. The other type is called the metabotropic receptor. These are a little bit different because there's an extra step involved. There are 3 steps with this metabotropic receptor. Neurotransmitter, here's its receptor site, so it's going to bind. But before the ion channel can open, there's something that needs to happen within the cell. That involves the neurotransmitter binding, then the G protein binds, then the ion channel opens. The advantage, something about the G protein allows it to respond to a wider variety of neurotransmitters. Do you see where we've got the cell body and the dendrites? We call that the input zone because it's taking in information. Why do we call the dendrite and cell body the input zone? What do they have? Receptors. That's why they are the input zone. Are you guys following? It's on hillock. Why is that important? Because the action potential is generated from there. That little area is called the integration zone. And then from the axon down is what we call induction zone, passive and saltatory, just the movement of the action potential down the length of the neuron. And then the very end is called the output zone. So these would be the axon terminals. So the little vesicles, they move, they fuse, they release. This is the output zone. You'll notice some of the cell body is part of the integration zone. The axon hillock, which incorporates some of the cell body and a little bit of the axon coming out. Then the conduction zone, then the output zone. Part of the cell body and the axon hillock have to decide, what are we going to do with this? So their job is to read the information, and this information is go. So this axon hillock now knows, hey, I've got to send an action potential. That's what happens after binding. Sometimes information gets to where it needs to go. Say it's in the dendrite, binds to the dendrites, and there's the decision. The integration zone has to say, okay, let's read the message, and then we'll make our decision. So if the message, after binding, is to, hey, stop the message here, what's the axon hillock going to do? They're not going to send an action potential. Sometimes it's important to not have too many action potentials. Do you guys know too many action potentials can lead to seizures? Instead of calling them go and stop messages, they've got names. After binding, messages are either inhibitory or excitatory. So an inhibitory message is don't send the message any further. Other one, the go message, it's an excitatory message. It is to go ahead and trigger an action potential. Inhibitory messages are actually called IPSPs. Stands for inhibitory postsynaptic potential. Postsynaptic, this tells you binding already happened, and now we're figuring out what to do now that we're on the postsynaptic neuron. This EPSP, which stands for excitatory postsynaptic potential. Right? That is Excellent. So when we talk about our frontal lobe, it turns out that we have a lot of inhibitory messages that are supposed to be binding in our frontal lobe. If for some reason they're not binding enough, or they're binding but they can't really do their job. So when we talk about poor impulse control, we could say maybe there's too many EPSPs, or we can say there's not enough IPSPs. But does the combination of these two, can it influence impulse control? The answer is absolutely. Okay. So again, these postsynaptic potentials, they're either inhibitory or they're excitatory. Different parts of the brain have more or less of those. Yeah. This is what we call neural integration. This, I really don't want to make this hard. It's not. Tons of neurons synapsing with this one neuron. This one neuron is getting information, let's just say, from 20 different neurons, and decide if it's supposed to fire or not fire. And by that, I mean create an action potential or not create an action potential. This neuron has a lot of decision-making to do. So the neural integration is the decision a neuron has to make based on all the information it's taking in from the synapsing neuron. We have what's called spatial summation and temporal summation. This is not from your textbook. What you have is you have a particular neuron that we're considering and all the input that it's getting. It could get input from different neurons, which we call spatial summation, or it can get lots of input from the same neuron, which we call temporal summation. When we talk about the brain requiring all this energy, this is what it's doing. So if this neuron just can't do it, it can't make the decision because it's getting totally overwhelmed, that's going to impair your function. So neuromodulation in general is when other chemicals and surrounding neurons can influence what goes on in a synapse. What we're saying is sometimes there are other things that can influence what goes on in the synapse. Sometimes other chemicals can tell it to release more neurotransmitters. Less neurotransmitters, it can affect binding. It's when something else outside is going to influence what goes on in the synapse. The one type of neuromodulation highlight right now is called an axonic synapse. Do you see this? What is that supposed to be? Axon terminal. Holy crap, we've got an axon terminal that's creating a synapse with an axon terminal. Axonic synapse. Why do we care about it? It can influence this.